Assalamu alaikum. I am Dr. Sania. Today we will study the development of the skeletal system. For the skeletal system, first of all we should know that what are the derivatives of the skeletal system. These are from the mesoderm and ectoderm. From the mesoderm there is the paraxial and the lateral plate mesoderm which will participate in the formation of the skeletal system. And in case of ectoderm, neural crust cells will participate. So, how the bones will ossify? We know that there is two types of ossification: intramembranous ossification and endochondral ossification. In case of intramembranous ossification, bone formation in which the mesenchyme differentiated directly into the bone, for example, flat bones of the skull. In endochondral ossification, it is also the bone formation process in which the mesenchymal cells give rise to the cartilaginous models first, which is then become ossified and form bone, for example long bones of the limbs. In this diagram you can appreciate the mesenchyme. Firstly, the cartilaginous model is formed. After that the ossification starts and blood vessels beats and the proliferation of chondrocytes takes place. And here you can also appreciate the epiphyseal cartilage. There are normally two centers of ossification: primary center of ossification and secondary center of ossification. Primary center of ossification is located in the shaft, or you can say in the diaphysis of the long bones, and the secondary ossification center is present at the ends of the long bone, as you can see here. So, development of the skull, we have the neurocranium and the viscerocranium. The neurocranium is a protective case for the brain, while the viscerocranium is the skeleton of the face. Firstly, we will discuss the neurocranium. Membranous neurocranium. This is formed by the intramembranous ossification. Mesenchymal cells are derived from the neural crust and paraxial mesoderm. Cells then encircle the brain and form most of the flat bones of the skull. You can see here in this diagram, these are some sutures: coronal sutures, sagittal suture is shown, and fontanelle are shown. Then you can see here again the posterolateral or the mastoid fontanelle and interior fontanelle or sphenoidal fontanelle. This one is the lymphoid suture. So, in the case of the neurocranium, it is developed from the mesenchymal cells which are derived from the neural crust cells and the paraxial mesoderm. This was the role of the membranous ossification. Now comes the cartilaginous neurocranium, the part of the neurocranium which is cartilaginous, also known as the chondrocranium. This is formed by the combination of mesodermal sclerotome and neural crust cells, and the cartilage are formed around the brain beginning at the notochord. Paracaudal cartilage and the occipital sclerotomes fuse to form the base of the occipital bone, while the sphenoid and ethmoidal bones are formed from the hypophyseal cartilage and the trabeculae carnae. All these pieces of bones fuse with each other to form a strong base of the skull, except for the except for the openings via which the cranial nerves leave the cell. So, we discussed that in case of the development of the skull, firstly we have the neurocranium and the viscerocranium. Neurocranium was a protective case for the brain, while the viscerocranium is the skeleton of the face. In case of the neurocranium, firstly we have the membranous neurocranium, where we discuss the role of the mesenchymal cells which were derived from the neural crust cells and the paraxial mesoderm. Then we discuss the cartilaginous neurocranium, which form by the combination of mesodermal sclerotome as well as neural crust cells. Now comes the viscerocranium. Again, membranous viscerocranium. Its dorsal portion undergoes intramembranous ossification and gives rise to the maxilla, the zygomatic bones, the squamous temporal bone, the vomer, and the palatine bones. So, all these bones will develop through the intramembranous ossification. In this diagram, you can see the maxillary process. Then we have the ventral portion of the viscerocranium, which contains the Meckel's cartilage. This region becomes surrounded by the mesenchymal cells that condenses and ossifies by the membranous ossification to form the mandible. So, in case of the ventral portion, the development will be of the mandible. It is also by the membranous ossification. Then we have the chondral viscerocranium. Its dorsal portion will develop the malleus and the incus. They form the stapes and the styloid process, Richard's cartilage. And the ventral portion ossifies and forms the lesser cornua and the upper body of the hyoid bone, while it also forms the greater cornua and the lower body of the hyoid bone. In this diagram, you can appreciate the zygomatic bone, the maxilla, mandible, Meckel's cartilage. Then we have the hyoid bone, thyroid cartilage, cricoid cartilage, malleus, stapes, and incus. Then comes the development of the vertebral column. Vertebral column originates from the sclerotomal cells. During the 4th week of development, these sclerotomal cells form the somites, which surround the ventromedial aspect of the notochord to form the centrum of the intervertebral disc. They will surround the dorsal portion of the neural tube to form the neural arch and also surround the ventrolateral aspect of the body wall to form the costal processes. This is very important to know that how the vertebral column will develop. First point to learn is which is important is that it will originate from the sclerotomal cells, and how this will proceed that these in the 4th week of the development, these sclerotomal cells from the somites will develop, and these from the somites they will surround the ventromedial aspect of the notochord, dorsal portion of the neural tube, while the ventrolateral aspect of the body wall. In this way they will form the centrum and intervertebral disc, neural arch, and costal process. Chondrification begins in the 6th week. Ossification begins before birth and end during the 25th year. At birth, 3 primary ossification centers are present in the centrum and in each half of the vertebral arch. This is the diagram in which you can see here the midline notochord and intersegmental mesenchyme. Here you can also appreciate the artery, nerve and the sclerotome segments. In the center you can see here the nucleus pulposus is developed. Then precartilaginous vertebral body is developed. Transverse process, annulus fibrosus, and ultimately the intervertebral disc get ossifies and it is formed. Ribs are derived from the sclerotome portion of the paraxial mesoderm, which form the costal process of the vertebrae. Costal process derived mainly from the thoracic vertebrae. Primary ossification center appear in the body of the ribs and mostly become cartilaginous during weeks 13 to 14 of development. Secondary ossification center will appear for the head and tubercle of the rib at puberty. Now comes the sternum. Sternum, it develops from the somatic mesoderm in the ventral body wall. There are two sternal bars which are formed on either side of the midline, and these later fuse to form the cartilaginous model of the manubrium. Sternum, which is the body, and the xiphoid process. Ossification appears cephalocaudally before birth, except in the xiphoid process which appears during childhood. So this is the point to note it, that xiphoid process ossification will appear during childhood. In neonate, the manubrium contains usually one main ossification center. Ossification at the lower segment begins shortly after birth and that of the xiphoid process during the 3rd year of life. Now we will discuss the appendicular skeleton. The limbs are derived from the somatic layer of the lateral mesoderm. Mesenchymal cells of this region become activated and the limb buds become visible as an outpocketing. Mesenchyme destined for the limbs is covered by a layer of ectoderm. Ectoderm thickens and forms the apical ectodermal ridge, which exerts an inductive influence and initiate growth. In this diagram you can see here, this is the ectoderm, overlying ectoderm, and this is the apical ectodermal ridge. In this diagram again, this is the apical ectodermal ridge. In the appendicular skeleton, distal end of the limb bud become flattened to form the hand plates and the foot plates. Fingers and toes are formed when the mesenchyme of the hand plates and the foot plates condenses to form digital rays by apoptosis. Similarly, as the shape of the limbs is being formed, mesenchyme in the buds condenses and differentiates into the chondrocytes. Entire limb skeleton is cartilaginous by the end of the 6th week of development. Joints are formed when chondrogenesis is arrested and a joint interzone is induced. In this diagram you can see here, with the red is shown the cell death area. These are the areas where the apoptosis is done, so the digital rays are here, and this one is with the green is the digital rays and the visible tissue, and the webs are formed here. Development of the upper and lower limb is similar except that the upper limb appeared approximately 1 to 2 days ahead of the lower limb. Upper limb birds develop opposite the cervical segments, while the lower limb birds form opposite the lumbar and upper sacral segments. End of the embryonic period, primary ossification begins in the diaphysis of the long bones. Endochondral ossification gradually progresses from diaphysis of the bone toward the end of the cartilaginous model. This is the diagram showing the lower limb bones. You can see here the pubis, ilium, tibia, fibula, and these are the foot plate cartilages. Here again the web is formed, and you can see here the apoptosis in these interdigital rays. And now the metatarsals and tarsal cartilages all are formed. The shaft or diaphysis of the long bone is fully ossified at birth. The epiphysis is still cartilaginous, and secondary ossification center appear in the epiphysis of these bones. Persistence of the growth plates provide for interstitial growth in the length of the long bone. Periosteum provides for oppositional growth in the girth of the bones. Endochondral ossification advances on both sides of the plate, and finally the plate disappears and the epiphysis unite with the shaft of the bone when bone has acquired its full length. This is the diagram showing the epiphysis, growth plate, diaphysis. Again this is the growth plate, and here is the secondary ossification center. I hope you have understood. Thank you.